we can skip altogether the fetal tissue, so the neurons and dopaminergic neurons, or neurons that will soon become dopaminergic, taken from embryons. This was the very first attempt that has generated a lot of interest back in the days. And we have now a sense of the fact that this can help. They can make some differences, not necessarily good differences, sometimes also side effects. At the moment, we don't want to explore this option too much because of mainly because of the difficulties in obtaining the cells, but also because these cells often are implanted with spurious material like serotoninergic cells. And there is an idea that implanting dopaminergic as well as serotoninergic cells all at once might actually increase one of these long-term side effects, which is called runaway dyskinesias. So people that were part of large trials in the past are still around these days. I've seen myself a couple, and they might have uncontrollable dyskinesias. And that's why we try to stay away from this type of protocols. So for sure, where the field is going, and this is where most of the actions are at the moment, is what I mentioned earlier as floor plate protocols. These are protocols where we use different cells. We either use embryonic stem cells, or this is the latest approach, the so-called iPSCs, which stands for inducible pluripotent stem cells, which is basically retroengineering cells. So there's a much better understanding of the biology of the cells and the processes involved in the evolution from stem cells to differentiated cells. We have a better understanding of the signaling pathways, of the paracrine processes involved. So for this reason, it's been done about, more than 10 years ago, for the first time, an experiment where cells of any kind were retroengineered so that they could become anything. As you probably know, everybody has the same genetic content in every possible cell. So in theory, any cells has the machinery and the genetic content to be anything. These induced pluripotent cells are, for example, coming from fibroblasts. You might take the fibroblasts from the skin of a subject, and you can make these fibroblasts go back to a more indifferentiated state, and then you can guide their evolution not towards fibroblasts, but towards progenitors of dopaminergic cells. So this is very exciting. And actually, this granted whoever discovered this in Japan the Nobel Prize in 2012, if I'm not mistaken. So these floor plate protocols rely either on embryonic cells, stem cells, or iPSCs, and I mentioned what they are. And they're called floor plate protocols because there's a lot of engineering and a lot of modulation and adjustments put in place to make sure that these cells become prolific, they increase in the number, and importantly, they become dopaminergic neurons. And what's also interesting about these protocols is that they can also be frozen, and so they can be used off the shelf whenever needed. So instead of using fresh material with a very narrow therapeutic, very narrow window when these cells need to be taken and implanted, instead, when you freeze these cells, you can basically use them whenever you want, and it's much easier also from a feasibility standpoint. So these type of cells, the floor plate protocols, is where most of the research is going on right now. And there are many centers in the world. I can mention at least 5 of interest where this is going on. For sure, a lot of what we know about self-replacement therapy comes from the effort that was led by Cambridge and London, respectively in the UK and in Sweden, and also with the establishment of a consortium called G-Force PD. They really pushed this field forward, initially with embryonic material, and more recently with these new protocols, and they're very active right now. They just started a new trial. There are other important centers, for example, CIRA, C-I-R-A, in Kyoto, in Japan, where the iPS story began. And they're also, at the moment, involved in new research projects. There is another consortium called ISCO in Australia, in Melbourne, and they are using very interesting type of cells, which are cells that are not fertilized. So there are cells coming from blastocysts, and they are unique in terms of genetic content. And some people are also arguing that might be dangerous to implant in humans. But so far, I know of at least 12 subjects that were implanted, and it seems to be safe, but it's very early to say. Coming to North America, there are important centers working on it, and I will probably just mention two which are related to two companies with interest and investments in this field. One is Blue Rock Therapeutic, and this is where I have a conflict of interest, because Blue Rock is based in New York, but with collaboration from Boston and Toronto, where I'm based. And as a matter of fact, Blue Rock is the one company that is probably a bit more advanced in terms of trials. They just started a phase 2 trial, and I can tell you more about the results of the phase 1 trial if you want. And the other company, which is based in California, is Aspen. And Aspen Neuroscience is using iPSCs, so induced pluripotent cells, but with a very interesting approach, because they're using autologous cells, meaning that they will enroll people with Parkinson's. They will take their cells, they will make them from the skin, they will make them dopaminergic neurons, and they will implant them in the brain. This comes with an incredible opportunity, because we won't need to use immunosuppressant. These cells obviously will have the same immunogenicity of any cells in the body, and therefore they won't be attacked by the immune system. So they will be more liable and more prolific also. And that's for sure something that will be of interest in the upcoming future. And the FDA has just granted approval for the phase 1 that this company is going to do.